microscope? What is microscope?
A microscope is an instrument that is used to magnify small objects. Some microscopes can even be used to observe an object at the cellular level, allowing scientists to see the shape of a cell, its nucleus, mitochondria, and other organelles.
History of microscope.
Timeline of the microscope. 14th century. Spectacles first made in Italy. 1590. Two Dutch spectacle makers and father and son team, Hans and Zacharias Janssen, create the first microscope. 1667. Robert Hooke's famous Micrographia is published, which outlines Hooke's various studies using the microscope. 1675. Enter Anton van Leeuwenhoek, who used a microscope with one lens to observe insects and other specimens. Leeuwenhoek was the first to observe bacteria.
In 1903, Richard Zsigmondy invents the ultramicroscope, which allows for observation of specimens below the wavelength of light. 1932, transparent biological materials are studied for the first time using Fritz Ziernicke's invention of the phase contrast microscope. 1938, just 6 years after the invention of the phase contrast microscope comes the electron microscope, developed by Ernst Röska, who realized that using electrons in microscopy enhanced resolution. 1981, 3D specimen images possible with the invention of the scanning tunneling microscope by Gerd Binninger. Types of microscope. Simple microscope, compound microscope, electron microscope, stereomicroscope, scanning probe microscope. Microscopic morphology is the study of the size, shape, and arrangement of cells and other materials using a microscope. It can also refer to the internal structure of a material, such as the arrangement of atoms. There are many types of microscopes, each with its own strengths and limitations. The right microscope to use depends on the information you want to gather and the size of the feature you want to observe. Some interference microscopy, a technique that uses two beams of light to pass through a specimen. Fluorescence microscopy, a technique that uses fluorescent dyes to attach to specific cellular components. Scanning electron microscopy, a technique that uses a moving spot of electrons to scan an object and produce a three-dimensional image. Microscopic morphology is often used to identify and differentiate cells. It can also be used to reveal chemical differences in cell components, and to observe structural components like flagella, endospores, and capsules.